Okay. Hi, hello everyone. So here comes the EPDS webinar with Chayong again. So today we are going to talk about something regarding immuno-oncology. So we are going to share the strategy and trends of small molecule drug discovery in immunodiscovery, especially focus on the opportunities and challenges of STING, ENPP, and PAP related pathways. So I'm Junyao Li from Eurofins Discovery Taiwan, and it's my great honor to share our insight with all of you. So at the beginning, just a short introduction of Eurofins Discovery. So we are a global leading CRO in drug discovery field. So we have more than 10 labs all around the world, including in APAC Pacific and Europe and US. So we are focusing mostly in the in vitro drug discovery, like biochemical assay, functional assay, or protein chemistry. And also we have in vivo capabilities. So in Taipei lab, we have over 50 years extensive experience in pharmacology. And also we have a different expertise in drug discovery for small molecules, and recently also for large molecule biologists. And for the target-based assay, actually we cover most of the druggable targets. For example, like GPCR, ion channel, transporter, nuclear hormone receptors, and also we have like whole profile for kinase and the other enzymes, and a lot of different translational cell models. And also we perform in vitro admin tox and in vivo PK and PD studies. And under the accreditation, we got like ALOC and OLOG accreditation. And also our operations under the structure of ISO 9001 and ISO 17025. And also we are supporting Japanese authorities for our Japanese clients issuing R&D to Japanese government. So this is the outline for today's webinar. So we are going to talk about the current oncology drug discovery strategies and trends. So we have our insights regarding this issue, and also we'll focus on the specific pathway called the STING pathway. And there are some molecular targets like STING, ENPP, and PAP that will be also of our topic today. So this is the general idea about the current drug discovery strategy in oncology field. So as we know, there are two different directions. The first one is very traditional one, and we just target directly on the tumor cells. So like the well-known chemotherapy, target therapy, and ADC or some hormone replacement. So we like to directly induce cytotoxicity or suppress the growth of tumor cells on the tumor itself. So we say this is the direct strategy, target on the tumor cells. And gradually we also have another thinking, focus more on the tumor microenvironment. So we say this is an indirect strategy. So we are thinking to activate the immune system. So this is a so-called cancer immunotherapy or immuno-oncology. So we try to modulate the environment, microenvironmental tumor, like tumor vasculature. So we have strategy in anti-angiogenesis or extracellular matrix. So there are some kind of MMP inhibitors under development, and also something related to cancer-associated fibroblast. So if we can affect the microenvironment of tumor, also we can suppress the growth of tumor or even to kill the tumor by our immune system. So that is another thinking strategy, and we can develop the tumor therapy. So there are some steps in cancer immunity cycles, and also there are some potential targets for our opportunities. So coming back to the tumor physiology, we know this cancer immune cycle. So once the tumor cells die, they will release some small piece of part of the tumor cell. Usually we call this as antigens. And the antigen will be exposed to the dendritic cell or antigen presenting cells, we call APC. So the cancer cell antigen will be presented. And then the system will pry and activate the APC and T cells. And the T cell will traffic to the tumor site by the infiltration and penetrate to the endothelial cell and then go to the tumor sites. And then they will recognize the tumor cells by T cell, and then the T cells start to, after recognition, T cells start to kill the tumor cell. And then when the tumor cell die, again they release more antigens, and then we trigger this cycle. During this whole cycle, there are a lot of molecular targets could be the target for anti-cancer drug treatments, and people are looking for such kind of targets for their drug discovery. So for example, in the cancer antigen presentation, we have like TGF pathway, interleukin families, or some CD40 related targets. And priming and activation, we can have like CD family or interleukin families, and also the well-known PD-1, PD-L1 pathway. And for the trafficking of T cell to tumors, we have CX3 families and CCL families. And for the infiltration, also we know the VEGF or endothelin B receptors. And then during the killing tumor cells, also the PD-1, PD-L1 recognizing system works, as well the other like IDO, TGF-beta, and TIM3, the others. So a lot of target could be considered as druggable target when we develop the tumor immunotherapy. So there are some representative immunotherapies approved by US FDA. So like the anti-CTLA4, anti-PD-1, so they are very popular and famous. And also like the other CAR T cell therapy is also under investigation, and also some cytokine modulators or oncolytic viruses. So a lot of different methods could be taken into consideration when we are thinking about the tumor immunotherapy. So from the market perspective, we can see the top 10 leading cancer drugs by their sales revenue. So of course, the top one is Merck Keytruda, and also we have the others from BSM, Janssen, and AstraZeneca. So among the top 10, we know 3 of them belong to the PD-1, PD-L1 antibody drugs, and 6 of them they are involved to the mechanism of immuno-oncology. So now we know the immuno-oncology has emerged as a dominant strategy in anti-cancer market. So our focus is shift from the direct pathway, direct target to tumor cells, to the indirectly to regulate the immune system, and to have some immuno-oncology strategy, and then can go to the anti-cancer markets. So there is also growing market in immunomodulators in cancer drug therapy. So in the next 5-6 years, still we can see the monoclonal antibodies still dominate the markets. But meanwhile, we can also see the immunomodulators, the proportion and the percentage will also grow in the coming few years until 2030. So the percentage we can see, even though the monoclonal antibodies, they will dominate, but the immune modulators, they are playing more and more roles. So for the immunomodulators, there are some opportunities for small molecules. So why there's increasing interest in studying immunomodulators for cancer therapy? So there are some rationales. So we know currently, like the PD-1, PD-L1, the immuno checkpoint inhibitors, they are very successful to cure the tumor, but also they have a very limited fraction of patients. So even though we know here the immunotherapy is dominated by the monoclonal antibody like a PD-1, PD-L1, CTLA4, they have been proven very effective in tumor immunotherapy. However, the success is only limited to a certain part of or a certain fraction of patients. So according to the literature, we are talking about this effective rate of monotherapy is only around 10 to 25%, or in average, we can say overall it's around 20%. So there are some approaches utilizing immunomodulators that could also regulate in the immune system the response to show some complementary or synergistic effect when we are using the immuno checkpoint inhibitor. Meaning when we apply the monoclonal antibody for the immunocheckpoint. Meanwhile, if we can think about to add some immunomodulators, maybe they can show some complementary or synergistic effect to increase the fraction of a patient being involved. And that will increase the percentage of effective population and make the immunocheckpoints inhibitors more effective in the tumor therapy. So here are just a comparison between the monoclonal antibody, or we call the large molecule compounds, and small molecule compounds. So in these two slides, I would just like to emphasize the advantage and disadvantage, or pros and cons, between the large molecule and small molecules. So as we know, the large molecule, the monoclonal antibodies, they have a very high selectivity, or we say the affinity and potency, and they have a lower off-target possibility, and non-specific toxicity is also lower, and they can have a longer duration of their activities. But of course, they got some limitation, like the cost is high for the manufacturing, and also they have a very poor or even no oral bioavailability. That will highly affect the patient compliance. And also they don't have a very good tissue penetration. So like in the brain area or some tumor cell, they have a large size, so the penetration is poor. And even though we say there's a low off-target or non-specific toxicity, but still there are still some risks for immunogenicity to have some kind of unexpected immune response. So finally we say, as mentioned, even though they are proven very successful and effective, but still the successful case is only for a fraction of patients, as we mentioned, it's around 20%. So still there are the rest of 80%, still we need to think about the other strategy. And next one is we are talking about more the small molecule strategy and why they make small molecule complementary tool in immuno-oncology. So coming back to the benefit for small molecule, and also I think this is also many scientists we are looking for the small molecule strategy. So the advantage is very obviously we can see it's easier to be synthesized and also easier to be produced under large scope. So we can easily produce large quantity of small molecules. And they are showing high permeability to tumor microenvironments or penetrate the tissue barrier, such like blood-brain barriers. And usually they have a better or good oral bioavailabilities that could increase the patient compliance. And they can cross the cell membrane. So not like large molecules, they can also only working on the cell surface. So for small molecules, they are able to penetrate cell membrane, and so they can both regulate extracellular and intracellular signal and targets. So that's why we are thinking the small molecule, there could be a complementary or synergistic effect when we combine with large molecule immunocheckpoint therapy, and they can have a synergistic effect. So later we'll explain more. Yeah, so this could be the next generation strategy for cancer immunotherapy by combining large molecule immunocheckpoint inhibitor and also small molecule complementary compounds. So there are some immunoregulatory targets already be developed or some presentative small molecule using or under clinical trial. So when we are seeing the microenvironment and some immunomodulators, so there are some kind of metabolites or some inhibitory cytokines. So here we can see some examples like CSF1R receptor inhibitor, or like a glutamate inhibitor, adenosine 2A receptor inhibitor, or like IDO1 and AHR inhibitors. And in the other way, also there are some targets to innate immune pathways, such like toll-like receptors, STING, and also some immunocheckpoint like a PD-1, PD-L1, MET4K1, CBLB inhibitor, or SHIP2. They are already very popular. So today we are going to talk more detail about the engaging cytosolic nucleic acid sensing. This is an emerging therapeutic strategy in oncology. Especially here, we're going to talk more about the STING and PAP related pathway, and there are some of the compound already under clinical trial. So coming back to the pathway, and we know the role of STING is very important in innate immunity. So the full name of STING is a stimulator of interferon genes. So this is our body's first line to defense against pathogens and abnormal cells by sensing the cytosolic DNA. So we can image when the outside invaders like bacteria, like virus, or like some external chemicals, especially so some pathogens. So when they enter our body, we can think about like virus. They want to use the RNA or DNA to infect our cells. So in our body, we have the enzyme called cGAS, cyclic GMP synthase. They will catalyze this kind of external cytosolic DNA into cyclic dinucleotide, we call CDN. And the CDN is a key to activate the STING pathway. So once we know the external DNA will be catalyzed by our enzyme and form the CDNs, and our immune system can recognize, okay, something outsider is coming, so we activate the immune response as our first line defense. So why we think this is some opportunity for cGAS STING pathogen in cancer immunotherapy? So we have to come back to the tumor environment. So usually in our body, there are some mechanism to suppress our innate immune system, especially in the tumor microenvironment. So when we have the tumor, when we are doing the radiation therapy, chemotherapy, or the new ones like CDM, so such kind of thing we can stimulate the STING pathway. So letting our immune system be wake up to understand, okay, there are something outsider coming, so the immune system need to be activated. So when the immune system, the STING pathway is activated, they release a lot of interferons, and then they will activate the T cell and make the tumor from cold to hot. And T cells, and NK cells, they will recognize the tumors, and they do their work to kill the tumor cells. So that's why when we activate the STING pathway, we call the tumor cells from the cold status to the hot status, and later T cell to recognize them to kill the tumors. So this is very important to convert the environment from cold to hot, and STING is playing the role. So to activate STING pathway, so there are some STING agonist strategy under development. So as we just say, some CDNs or cyclic dinucleotide or the similar analogs, so they are under some clinical trial. But the CDN, they got some problems, later we'll show. So that's why also people are thinking about the non-CDN structures or even carriers or some antibody drug conjugates. And we are trying to bring the signal and specifically activate the STING signal on the target tumor site. And you can see here a lot of pharmas, they are working on the clinical trial, and usually on the phase 1 and 2, and to develop such kind of STING-related agonist. So the current challenging of STING agonist on immune oncology drug discovery are listed here. So the cyclic dinucleotide actually is similar to our physiological signals, so they are not very stable in vivo. So when we are thinking about the structure, maybe we try to modify the structure to make them not easy to increase their stability in vivo. And also the therapeutic window is narrow for the STING agonist. So sometimes they can produce kind of unexpected immune response or raise some safety concern. So for that, we are thinking about some carriers or antibody conjugates, try to more specific to the target area to reduce the unexpected effects. And also the third one, so this is very important. So there's significant heterogeneity and population diversities for the STING pathway. So for the common human STING variants, we have the wild type is called R232. But meanwhile, we have a lot of variants, or we call SNP, during the human population. So different STING variants, they could show very different response to the potency to the CDN or non-CDN across different STING variants. So not all the variants, they will be responsive to our small molecule compounds. And also they are, during the study, the rodent, especially for mice, clinical anti-tumor activity, because mice, they also have a different variants. So when we shift from mice to human, also there are some variant difference. So they will also make some disconnection between the clinical study to the human clinical studies. So considering of the genetic variation and species cross-reaction, this is very crucial to the STING drug discovery, and we need to make some strategy to ensure the clinical efficacy and safety. So this is just some example. We are talking about the single nucleotide polymorphism, or we call SNP, or a variant of STING. So we can see the most common human variants is almost 60%. It's our wild type of R232. But also we know like HAQ or R232H, R293 or R71, there are also some populations in the human society. And when we are looking for the different race, like American, Europeans, and East Asia, so there are still some different, even though they are most common is the wild type, but still there are other variants could be also very common. So the human race and human polymorphism variation could make the drug discovery strategy different. And when we are working some preclinical study and we want to apply to human clinical trial, we need to think about the variation between different polymorphisms. So here are just some in-house study and showing the example different responses from different variants. So the most common endogenous CDN ligand 2,3-CGAMP, we can see they show very consistent affinity and EC50 among all the variants except the R32H. So they show very low affinity compared to the other variants. And the right side we can see the newly discovered non-CDN STING agonist. This is a small molecule, diABZ. So they show very different response regarding the IC50 to different variants. So like to the wild type or G230A, they show very high affinity and low IC50 value. But like for the HAQ and the R293, they show less affinity. It's around 10 times different. So when we are discovering the STING agonist, or we are designing this STING agonist, we need to think about the variance of the different kind of human variants. So considering this kind of variation among the human variants, so Eurofins Discovery, we designed a panel for STING human variant. We call this STING human variant lead hunter panel. So we include the most common human variant from the wild type, G230, AQ, HQ, and R293. So there are different polymorphisms, and also we include the 2 animal species, including mouse and monkey. So we try to use this panel to cover common humans STING genetic variants, and they are cell functional assay. So we can validate the compounds by different variations, and we can provide the information regarding the human genetic variants, and also provide the different species. We can do the cross-species comparison. So such kind of panel will help when we are designing the compound. We can see the response between different variants, and this will be very important when we try to translate the animal study to human, or when we design the clinical trial. We need to understand, maybe for the patients we need to check their variants, when we design the clinical trial, we need to consider the final effect. Maybe they could be correlated to the human variant. So beside the STING pathway itself, there are also some peripheral targets. They are also very important to regulate the STING activity. So PAP7 acts a kind of immunosuppression or as a break of cytosolic cyclic nucleic acid sensing pathway. So in general, we can say PAP7 they inhibit STING pathway. So if we can think about it, we can inhibit PAP7 and then we can restore the STING pathway. So PAP7 inhibitor is also a kind of drug discovery strategy in this STING pathway. So this slide we show the PAP superfamily members. So here we know PAP7 is very, very important in regulating the STING pathway, so they are used or critical for the innate immunity. So they are used for some advanced solid tumor like lung cancer. But meanwhile, we know this other family of PAP, like PAP1 to 3, they have a function in DNA damage repair. And PAP5A and B, they show some maintenance function in the telomere and regulating cell signaling. So they are also very widely used to the cancer targets and could be used for different kind of cancer like ovarian, breast cancer, or some advanced tumor cells. So overall, there are 17 members of PAP family, but the most well-recognized one is for PAP1, 2, 3, 5A, 5B, and 7. So they play a very important role in DNA repair or maintenance of telomere or Wnt beta catenin signaling, and also as we know for the STING signaling pathway. And indeed, there are some small molecule compounds under discovery or even clinical trial, and one compound is already approved by TFDA. So here we can see the Oraparib, they are TFDA approved compound targeted on PAP1 and 2. Another one, Pamiparib, for ovarian cancer is also approved in China, targeted on PAP1 and 2, and showing very high affinity. And another one for PAP7, as we mentioned today about STING pathways, RBN 3297. So they are under phase 1 and 2 clinical trial. And the other one, we say the PAP5A and B, we have a compound XAV and ME compound. They are also designed for prostate cancer and breast cancer. So the PAP family is also very important, and this is a cancer therapy strategy for developing the PAP-related small molecule inhibitors. So according to the scenario Eurofins, we also design a PAP screen panel. So we call this is PAP screen redundant panel. This is a quick panel to evaluate the selectivity of PAP inhibitors. So once we have an inhibitor on PAP, we want to know their selectivity across different subtypes. So we can use this panel to check their selectivity. So right side we can see the data, RBN 2397, this is a selective PAP7 inhibitor. So we can see they show very low IC50 or high affinity compared to the other subtypes. So that could help us to screen out the potential or selective PAP inhibitors on different subtypes. And the last one, we are going to talk about the EMPP pathway. So we have a STING pathway, and we have some PAP7, they will inhibit STING pathway. And extracellularly also we have an enzyme called EMPP. So EMPP is an extracellular enzyme that negatively regulates STING pathway by hydrolyzing extracellular 2,3-C-GAMP. So they will, EMPP itself can increase the C-GAMP and also convert the C-GAMP to AMP and also convert them into adenosine. So they have the effect to suppress the immune system. So if we can design inhibitor on EMPP1, and that will enhance the anti-tumor immunity via increase the extracellular C-GAMP level, and we know C-GAMP will activate STING pathway. And also we can reduce the synthesis of adenosine. So there are some demonstrative data of clinical candidates for our reference. So we know EMPP is also a family, and EMPP1 here we can see like the compound EMPP1-IN1. This is used for cancer immunotherapy, and they show very high affinity EMPP1. But also in the EMPP family, like EMPP2, there are also a lot of different compounds. They show very high affinity to EMPP2, and they have some therapeutic potential for fibrosis. So this family is also very well recognized for some kind of like a cancer immunotherapy or some anti-fibrosis. So if we can recognize the selectivity between EMPP1 or 2 or the other families, that will be also provide some very useful information when we discover the EMPP related compounds. So based on the background, Eurofins also designed the EMPP screen lead on the panel to include all the EMPP families to help our client when we design the EMPP compounds, especially on EMPP1 and 2. We can check the selectivity across all the subfamilies. And we are also the first CRO offering the whole EMPP families for the screening purpose. And this is a very cost-effective way to evaluate the selectivity across EMPP 1 to 7. And so from the right side, we can see the reference compound EMPP1-IN1 show over 200-fold inhibitory potency against EMPP1 to the others. So the selectivity could be observed when we have some small molecule compound be screened by this panel. So in the end, we would just like to share our insight and some take-home messages for your information. So small molecules still hold a very great potential in immunomodulation, and the complementary or synergistic effect could be expected when we combine with large molecule compounds, especially for the immunocheckpoint inhibitors like a PD-1, PD-L1, CTLA-4. So they show some potential in combination. So that's why the small molecule complementary therapy is still a very hot topic. And among this strategy, small molecule strategy, the C-gasting pathway is an emerging therapeutic strategy in oncology. But when we are thinking about this pathway, we need to also consider the genetic variation between species and human variants. So this is very crucial when we are doing the translation from animal to human, and also during we are going to the clinical trial. And not only for the stem pathway itself, but also some peripheral targets, such like EMPP1, PAP7, they could also offer some opportunities to enhance the anti-human immunity. So this is the key message we are going to share today. And this is some summary of the opportunity for small molecule in cancer immunotherapy. So like STING pathway, there are some different kind of variants. And PAP, there are some subtypes. We know the PAP family, they are widely used for cancer drug discovery. And EMPP, especially EMPP 1 and 2, they have more therapeutic usage. And not only STING, PAP, and EMPP, also some other small molecule IO targets, as well-known PD-1, PD-L1, CTLA-4, CD80, or even adenosine receptor, EP2, EP4 receptor, or COX. Or here, right side, we can see some others like IDO, AR, or CXCR family, TGF-beta. Such kind of molecular target could be also some interesting targets for the cancer immunotherapy. Then also Eurofins, not only Taipei side, but in the US side, we provide different kind of translational cell models for cancer immunotherapy. Like we have a biomap, or we have a cytokine release assay from human PBMC. They can also help us to evaluate the immunotherapy potential for some potential compounds. So Eurofins Discovery, we are trying to help our client and support their oncology drug discovery. So we provide one-stop oncology service in Eurofins, from target-based assay, as we mentioned, like the hard targets, STING pathway, EMPP, PAP, or even some conventional pathway like nuclear hormone receptors, growth factors, or kinase. Like in Eurofins, we have more than 600 different kind of kinase assays. So that could be provide some information on the target-based screening. And after target-based screening, we know the molecular targets, and then we can go through the cell-based assay. So in Eurofins, we have a tumor profiling panel. We select 36 tumor cancers corresponding to xenograft and synergic model in vivo, validated by in vivo efficacy study. But if we want to more different kind of tumor types, we have onco panel and showing more than 300 different kind of genomically diverse tumor cells. And also we have some kind of a phenotypic cell-based assay, like ADCC, CDC, biomap, or onco panels. And after the in vitro cell-based assay, we can go to the final in vivo models. So in Eurofins PDS, we have more than 60 different kind of validated animal models for oncology study. And meanwhile, we can also provide PK studies for these anti-tumor drugs. So thank you very much for your attention, and please don't hesitate to raise any questions. We are very glad to help your drug discovery journey. Thank you.